Hi everyone, I'm Brooke. So my protein is CROSS. CROSS is a relay molecule, so it's located on the inner side of the plasma membrane where it can interact with internal cell membranes. It's related to various cancers since its main function is regulating those cell signaling pathways that control a variety of cellular processes such as cellular growth, differentiation, migration, and survival. So now moving on to the structure of the CROSS protein, I'm going to start out with explaining its primary structure. So the primary structure is responsible for illustrating the linear amino acid sequence of the protein. So I'm going to represent that here with these beads. So each bead here is representing a different amino acid. So here, the blue one, I'm highlighting that glycine amino acid, and that is found in the normal CROSS protein. So then moving on to the secondary structure of the CROSS protein, it is composed of alpha helices, which I have here, they're nice and tightly coiled, as well as those beta sheets. So the tertiary structure of CROSS involves folding multiple of these alpha and beta helices together into a compact globular 3D shape here. This is where the functional domains of CROSS are formed, such as the GTP binding site, which I'm going to talk about in a moment. The overall shape is influenced by those R group interactions. As I mentioned previously, CROSS is a relay molecule playing an important role in the signaling, growth, survival, regulation, all of those. So when CROSS is bound to GDP, it is in its inactive state, and no signals are relayed to the cell's nucleus. CROSS must be bound to GTP to become active and transmit signals to various downstream pathways, including those involved in the cell growth, proliferation, and survival. So in the case of the G12C mutation in the CROSS gene, this mutation occurs specifically at codon 12, which is the GTP binding domain. So in normal CROSS, the sequence is GGT, and that codes for glycine, which I showed in the blue. However, in the mutated version, the G changes to a T, which then alters the amino acid that's being encoded. So TGT codes then for cysteine, which now I'm showing in this purple as the cysteine. So this change in the amino acid sequence has significant effects on the CROSS's protein ability to function properly. So we're going to break it down. Glycine, which I showed originally in the blue, that's the normal, is small nonpolar amino acid, which has a simple hydrogen atom as its side chain. So this side chain, as I discussed earlier, contributes to that tertiary structure of the protein. Specifically, it's providing flexibility to the CROSS protein structure, allowing the glycine to bend and twist more easily. So this characteristic is important for the protein's ability to undergo conformational changes like binding to GTP in the protein's active form, as well as hydrolyzing that GDP in the inactive form. However, moving on to that mutated form contains a cysteine amino acid instead. So in contrast, the cysteine is a lot larger, more polar, sulfur-containing amino acid. That presence of the SH group in the cysteine allows it to form disulfide bonds with the other cysteine residues in the protein or with other molecules. This contributes to a much more rigid and restrictive protein structure. This rigidity interferes with the protein's ability to undergo conformational changes. Specifically, this mutation of changing a single nucleotide base pair in the DNA sequence from that glycine to the cysteine then creates a structural block that prevents CROSS from transitioning from its active to its inactive state. So specifically, cysteine locks the protein in the active state because it doesn't allow the protein to reset properly by hydrolyzing that GTP. So staying in this GTP bound state causes the CROSS protein to remain on and continuously perform that function of cellular growth. So as a result, uncontrolled cellular proliferation occurs, which is, as many of you know, a hallmark of cancer. Overall, this is just a model, these things that I showed you, so it's very simplified and doesn't fully capture all of the interactions that CROSS has. So for instance, CROSS interacts with other proteins that I didn't show, such as GAPs and GEFs. In addition, CROSS undergoes important conformational changes when it binds to GTP versus GDP. And the G12C mutation specifically disrupts these changes. And my model doesn't fully capture those dynamic conformational shifts that are crucial for the cross protein's function. Also, I showed that mutation by changing the color of the bead at that codon 12 and changing those amino acids. But more ideally, the beads would be a different size. To specifically, the cysteine would be a lot larger, contributing to that rigidity that I talked about, whereas the glycine would be a lot smaller with the more flexibility, in order to show that mutation more realistic in the protein. But that is all. Thank you so much. Hope you got to learn more about cross.